Okay, so we talked a little bit about an introduction to cancer. You compared a normal cell's function to what we're going to look at for cancer cells, misfunction or malfunctions. So here's kind of the big picture. When you're looking at the genetics of cancer, what you really want to remember is that cancer is primarily caused by mutants, so mutations. And typically with cancers, it's because of progressive mutations, not usually one mutation, but several. Another thing to look at is that you can have cancers that have a tendency to be hereditary, or you can have cancers that spontaneously occur. So you want to consider some of the research that's been done, some of the historical research. And a lot of the popular research that says that there's an environment that has a lot of effect is based on Japanese immigrants. So we already know that Japanese women have a lower rate of breast cancer. So something good about their genes that makes them less susceptible to get breast cancer. But if you take the same Japanese heritage, a woman's born in Japan, and you raise her in the United States, it increases her risk for breast cancer. So there's something about her environment that's actually changing them and affecting them and causing cancer. And it's the same thing with men and prostate cancer. So prostate cancer in a Japanese-born male is lower, but you take that Japanese-born male and bring them here to the United States, and it increases the risk of prostate cancer. So the big question is cancer caused by hereditary or your lifestyle? And the answer is kind of both. So it's nature versus nurture. So there's some nature tendencies that may lead you or make you more prone to get a cancer, but there's a lot of environmental influence. We're going to talk about those too. I love this picture. If you're going to be dumb, you better be tough. And I kind of think about that with the things that we do, like smoking or excessive drinking and doing things that we shouldn't be doing, putting things in our body or exposing ourselves to things we shouldn't. If you're going to be dumb and do all those things, you better hope you have really good genes. I love this guy's Army shirt. I'm sure he was probably never in the Army, and he definitely doesn't do them justice. But this is another one that always kind of like shocks me. But this one reminds me of my grandmother, Cancer Schmancer. So I think I already talked about this picture before, but it's interesting because when you look at it, some people that smoke their entire life and never get cancer versus some people that smoke for a year and get cancer. You know, it's just not predictable. You can't predict who's definitely going to get cancer. There's no guarantee. There are a lot of factors that go into it, and we're going to talk about a lot of those factors that revolve around your genetics. So first, there are a couple of rules. And you want to think back about the normal function of the cell. Cells respond to things called growth factors. So they reproduce because of outside signals. They have to have receptors for those signals. Once those receptors are turned on, there's a pathway called second messenger system, a cascading pathway like dominoes. They're a chain reaction that go from out here all the way down and turn something on that makes the DNA copy, produce new proteins, possibly make new receptors, new structures or functions, or new enzymes. So there's kind of some general rules here, but cancer affects gene expression. So it turns something on or turns something off, and unfortunately it's turning the wrong thing on or off, and we're going to talk about those examples. So like I said, it can be familial or it can be sporadic. And familial, a really good example that I'm sure you're all familiar with, is breast cancer. So we've looked at breast cancer is one of the most common hereditary cancers. And the most common gene that's affected are called the BRCA genes, or people call them BRCA genes, but the BRCA genes. And what they find is that these BRCA genes, if you have a mutant or a defective one, you increase your risk for breast cancer. So men or women can carry this gene. We know exactly where the genes are. What happens are those genes are there to actually stop. They're called tumor suppressor genes. They stop a potential cancer cell from replicating. But the problem is if you have this broken, then you can't stop it from replicating. And it goes on to replicate, and it can cause a mutated cell to replicate, which can lead to cancer. And here you can see some overall statistics. We find that if you have the BRCA gene compared to the normal population, this is their rate of cancer, breast cancer, where somebody that has BRCA, here's their rate of cancer. So you can see that BRCA gene actually increases the risk of getting a breast cancer. But what's interesting is that only about 10% of the 180,000 breast cancers that are diagnosed in the United States each year, only 10% of those actually have this flaw. So you have to kind of look at the information and step back from it and say, does this cause it? Well, it increases your risk, but it doesn't necessarily cause it. If it caused it directly, then every one of these women with the BRCA gene would have breast cancer. So it's not always a direct cause, but it does lead them to be more susceptible. And another thing that it tells us is that there are people out there getting breast cancer that don't have the BRCA gene. In fact, 90% of people that get breast cancer don't have the flawed gene. So that tells us there are a lot of other causes too, which makes cancer really hard and difficult to work with. So does a bad gene ensure that you're going to get cancer? I mean, we just figured that out. No, it doesn't. There are a lot of factors. In fact, what's interesting is that we looked at this in genetics, but the genetic epigenetic changes that happen. When you're talking about a genetic change, it's actually the DNA that's been changed. When you're talking about epigenetic change, the DNA is exactly the same, but the gene expression has changed. And you learned about that from one of the homework questions in the last section, from the genetics section. So it's interesting because it could be situations like this. Maybe you have the flawed gene, but something about epigenetics, something you're doing to your environment or some environmental criteria has changed it so that you don't express a flawed or bad gene that could cause cancer. And you really want to think of that situation like penetrance that we talked about before too, where some people that have the bad gene don't necessarily get the disorder. The penetrance example last time was, which one was that? Oh, retinoblastoma, where just because you get the genes for retinoblastoma, only about 90% of those people will get retinoblastoma. Not everybody gets it. So there may be some epigenetic influence in there. So the moral of the story is just because you have the bad genes, don't give up and just think that you're doomed, that this is definitely going to affect you, because it may not. Right? If you want a little bit of a refresher on epigenetics, this is kind of cool, and this is just an extreme overview. But here you have your chromosome, your chromatin. All of your DNA is wrapped around these little things called histones. Well, these histones, when you need to copy them to make the gene, you have to unwind them. But just one of the forms of epigenetics may keep you from unwinding around that histone properly, so you can't read the gene, which means you can't use the gene. My analogy in my head is that, let's say you have two blenders. You have a really nice blender, a good blender. But what happens if the cord gets all tangled up and you're in a hurry? Well, you just grab the crappy blender, right? Because it's easily accessible. The same thing with these genes. Maybe something in your environment has tangled this up, an epigenetic change, tangled it up so you can't open up that gene. So you just use your other copy of it. So that's my simple analogy. If you want to go and look at epigenetics in a lot more detail, I actually put a link here. I love this little page because it's a really good overview. And it's from the Scientist magazine. And here's the link that you could actually find the picture and blow it up if you want to, in case the picture that I sent you doesn't come out very clear. But it's just a primer overview of generally how epigenetics works. So there's one factor you have to keep in mind, that just because you have the gene doesn't mean you're doomed to a specific destiny. Right? Another thing is most cases are actually sporadic. So most cancers are actually sporadic, which means they just happen because of mutations. And there are different types of mutations we talk about in genetics. There are point mutations where it's just one single letter in here that changes. You can also have chromosome aberrations or alterations where an entire section of a chromosome may break free, flipped around and glued back in, or rearranged and glued back in. But we're going to talk about some of these different factors. And we're going to talk about some specific types of mutations that happen and what kind of genes they mutate to or mutate from. So here's an example of a normal cell. Here you have some, well, in this example, it's a clastogen, because remember, a clastogen breaks chromosomes. So it broke the chromosome, flipped it around, your repair mechanisms glued it back into place, but inappropriately, so now that gene's not expressed properly. Now that cell can mutate, and then it causes all these other replicated mutated cells that are just as effective as the first, because they copy the chromosome exactly, and now the copies in these new replicas look exactly like the original. So some terminology or some things you need to remember. When I use these terms like checkpoints, proto-oncogens, tumor suppressor genes, and repair enzymes, I want you to remember what these mean. A checkpoint, when we're talking about the cell cycle, when the cell's getting ready to replicate, it goes through the cell cycle. Now remember, normally it's in something called G0, which means it's not growing. Most cells are like that. But when they're ready to grow and replicate, like to fill in a cut or a wound, or to make new tissue or grow something new, they go through this G1 phase, or this growth phase, where they're rapidly growing. They're getting more and more raw materials inside. They're starting to replicate things like organelles. And then they come to this checkpoint. And the checkpoint's kind of like a toll booth. You pull your car up to it and you stop, right? And then let's say it's a drug toll booth. They're going to search your car, make sure everything's okay before they let you pass through. So you've got this little thing called a tumor suppressor gene, that if your car's not safe, or your cell's not ready to move on, this tumor suppressor gene will hold it there. It won't let your car pass. It won't let your cell pass this point. It's stuck. Then a repair mechanism comes in. So these little things called proto-oncogens usually make things that help repair or fix or increase replication activity. But let's say this repair mechanism comes in, it's going to fix your car. Now your car is all ready. This tumor suppressor shuts off, and then zoom, you go moving along again. You keep cruising along until you hit the second checkpoint. When you hit that checkpoint, it's the same thing. You stop, you check to make sure everything worked fine. If S phase, when you're synthesizing DNA, if that DNA is flawed, there's problems with the DNA, there's defects in the DNA, let's say your organelles didn't replicate appropriately, if you're not really prepared at this point, then what happens is you go through this repair mechanism. Fix the DNA. You double check it at the checkpoint. If it's still no good, then what will typically happen is the cell will go into apoptosis. So it dies. It's flawed. It doesn't want to make any replicas, so it just dies so another healthy cell can replace it. But if the repair mechanisms work, then you proceed through this. So you just keep sliding right along. Then you go into mitosis, and that's where the cell actually replicates. And it starts replicating, moving along. It's going through prophase and metaphase, and then it's getting ready to replicate and split, and before it splits, it goes through one last checkpoint. And wants to make sure that everything's dividing appropriately. Did the mitochondria divide appropriately? Did the organelles move to the right areas? Did everything move appropriately? And if it doesn't, then it goes to apoptosis. If any of these checkpoints fail, the cell dies naturally. It knows it's flawed, it dies. But if you can fix it, then it goes through cytokinesis, it divides, and then you have these two new cells. So those are just some terms to get familiar with. The checkpoints are these little stop phases that you want to make sure the cell's ready to replicate. Proto-oncogens increase, they're like accelerators. They push the reproduction. They speed things up. The tumor suppressor cells try and stop it. And if tumor suppressor cells can't stop it long enough to repair the cell, then the cell goes through apoptosis and dies. And then the little repair mechanisms are the things that come in and try and fix the DNA and straighten things out and organize it, right? The term proto-oncogen, when you see onco, usually you probably think cancer, but a proto-oncogen is actually a good gene. It's good. It's helping the cell replicate. It's helping proliferate, that term. It's when it becomes something called an oncogene. When it's flawed, then it's actually bad, and oncogenes lead to cancers or tumors. All right, so let's go through some of those more specifically. So first we'll talk about those proto-oncogens, accelerators. When you look at a cell on the outside, you have receptors that are sensitive to different chemicals. Inside you have these second messengers. The second messengers turn on DNA and cause replication. When you have a proto-oncogen flaw, there can be a flaw in any of these steps. You can have this receptor stuck open. You can have too many of the hormones stimulating it. You can have any of these pathways stuck on. You can have the DNA that's stuck in the on replication position. So these are all flaws. So too many growth factors out here. A flaw with the receptor, it's stuck in the on position or mutated the on position. Or you can have these intracellular proteins that are constantly turned on. They're not waiting for a signal out here. This receptor could be completely off or missing, but these signals keep going in. So basically the accelerator's stuck. And that cell's going to replicate uncontrollably over and over and over again. And that's the significance of a proto-oncogen, is they increase proliferation and replication. A lot of cancer cells will actually have proto-oncogens that become oncogens, and they're stuck on all the time. Right? Your book refers to it as pro-growth, but proto-oncogens. And a common one is actually the RAS. And I usually don't make you remember molecular names, but this is an important one. RAS is a second messenger in here that increases cell growth. If that gets stuck on, then your cell wants to keep replicating over and over and over and over again. And they found that these RAS mutations are common in things like lung cancer, pancreatic cancers, and melanomas. So they're very, very dangerous. In fact, one of the first diseases we'll talk about here, cancers, is called pancreatic ductal adenocarcinoma. So hopefully you remember adeno or adenocarcinoma. Carcinoma means that it's a what? A tumor or a cancer? It's cancer. Adeno means it's referring to what type of tissue? Epithelial. But adeno is specifically talking about what? The sheet-like epithelial or the gland epithelial? It's the glands. So here's what's wrong with pancreatic cancer. About 95% of all pancreatic cancers, they have this KRAS gene that's mutated, it's stuck on. All right? PDAC stands for pancreatic ductal adenocarcinoma. So anyway, 95% of these pancreatic cancer cases are going to be this PDAC. Right? And of course, it's an oncogen. Remember, this was a proto-oncogen before, but now that it's causing a cancer, we call it an oncogen. And what happens is that these cells that are mutated, now they're called cancer stem cells. And it's in the quotations because they're not real stem cells. They're actually cancer cells that are replicating as if they're a stem cell. So they keep replicating over and over and over and over again. Right? So that term cancer stem cells, you want to be familiar with this, not an actual stem cell. It's a cell that's acting like a stem cell because it's a cancer. Right? When you look at the survival rate of these, so this thing keeps replicating. It can grow a mass. Why do you think it's really hard for these to get diagnosed right away? Well, think about the location. It's deep inside of you. And since the pancreas is adjusting things like digestion, usually most of the symptoms are like maybe tummy aches, weight loss, malnutrition a little bit, some appetite loss because you just feel like you have an upset stomach. So they're really insidious symptoms that you don't think about them until they're too big. And then once that cancer is mutated enough, it starts sending out all these little other cancer cells to places like the liver or the stomach or even to the lungs or the brain. It starts metastasizing. By the time that it starts metastasizing, usually when the symptoms are so bad that people go and get checked out, and at that point it's really dangerous. That's one of the reasons pancreatic cancer is so dangerous, is because it takes so long to be diagnosed. So when you look, the 1-year survival rate of somebody that's been diagnosed with pancreatic cancer is about 25%. So 25% of those people will live for a year. About 6% will live for 5 years or more. So they're not really good survival rates. If you remember, Steve Jobs was diagnosed with pancreatic cancer, but he lived, it was like 8 years. So from 2003 when he was diagnosed until 2011 is when he passed. So 8 years he lived with it. And you may wonder, well, geez, is it money? Is it that's what did it? And not really. It's because he had a really rare form of pancreatic cancer. So he didn't have the 95% case. He actually had a different type. His was called neuroendocrine cancer, which is actually really slow growing. So once they diagnosed it, it was easier for them to treat, but it still took his life eventually, as you know. So the average survival rate for a person with pancreatic ductal adenocarcinoma is only about 6 to 10 months. All right, the next type of genes are the brakes. So if those proto-oncogenes are accelerators, then you need some brakes that are like the stop genes. And these are our tumor suppressor genes. Brain fart for a second there. But yeah, our tumor suppressor genes, the little stop people. So they're the brakes. So if this gets turned on too much, you've got brakes in here that will stop it and say, wait, we're changing too fast, we're mutated, we've got to stop and fix this stuff. So there are a couple of really common ones. One's called the RB tumor suppressor gene. We'll talk about this. It's called the retinoblastoma tumor suppressor gene. We talked a little about retinoblastoma and genetics, but we're going to talk about it a little bit more in detail. Right? So basically what happens is with these things, if you turn on this pathway and the brakes stop the pathway, the cell will stop, let you fix it. But if you inactivate a tumor suppressor, when you hit the accelerator, there's no stopping. It's like removing the brake and the emergency brake from your car. Right? Another really common one is the P53 gene, and we'll talk about that a little bit more too. So P53 does the same thing. P53 is actually doing a lot of stuff, and I have a whole slide devoted to it. But what these try to do is they try and stop. They suppress the cell from moving forward. So this is kind of like the cell without a tumor suppressor. It's just flying right along with nothing to stop it. So it's doomed. Normally, tumor suppressor cells, if they can't give you enough time to fix the cell, they will trigger something called apoptosis, and you know what that is. So apoptosis is cell suicide. When you turn off these tumor suppressors, you also turn off apoptosis, so the cell can freely replicate over and over and over again. And we've already talked about one, and it's called the BRCA or BRCA gene that causes breast and ovarian cancer. So here's an example, and we talked about this before too. So neurofibromatosis, or a neurofibromatoma. If you remember, the name of it was von Recklinghausen's syndrome, or disorder. And it's where these people get these fibrous neural growths that project all over their body. When we talked about it in the genetic section, we referred to it as being an autosomal dominant disorder, which means if you got just one gene for it, you get these. We also talked about this being expressivity, where some people express it more than others. Like some people might have those little cafe au lait spots, the little dark spots, and they look like birthmarks. Some people get these larger tumors that are growing all over their body, but they're benign tumors. They're painful, they're uncomfortable, but they're benign. And then some people have an extreme case, like this is Joseph Merrick. You probably have heard of him if you look at old legends and stuff, but Joseph Merrick was known as the Elephant Man, and he had neurofibromatosis. since we're talking about this with mutations now, about 50% of all of the cases of neurofibromatomas are sporadic mutations. So they're benign tumors that just happen. So you can refer to it as an autosomal dominant or sporadic mutation case in either situation. But what happens here is that they've lost the tumor suppressors. These cells just keep rapidly replicating and growing, and they bulge out of the surface. Right? We even know what gene it is. It's the NF1, so neurofibromatoma 1 gene. Right? Normally what it does is it stops RAS. It stops the accelerator. Remember, RAS is an accelerator, NF1 is the brake. So you can put your foot on the accelerator and put your foot on the brake at the same time, and your car shouldn't go anywhere because these tumor suppressors are powerful. But if you turn off the tumor suppressor, that accelerator will drive the cell. It just keeps making it go. Right? Next, the repair enzymes. So maybe you have a faulty repair enzyme. So let's say this repair guy isn't working appropriately. One example is this PARP. So poly-ADP ribose polymerase is what it is, but it's a repair enzyme. So here's your normal cell. Your normal cell is metabolizing, it's living, it gets a little bit of damage, whether it's from UV radiation or some toxins or something. It gets nuclear damage, mitochondrial damage. Your repair mechanisms come in, they fix it, and they bring it back to new again. And they just keep going through this. But what happens if the repair guy is not working? Well, you don't repair it, and then you get sick. Either the cell is going to recognize it's sick and can't repair itself, and it goes through apoptosis. Okay? Or it can just age and not replicate, or it becomes a cancer cell and then just goes crazy with replication. Right? So there are really 3 outcomes to what happens when a repair enzyme fails. But you can see PARP here. You have a DNA break, just one of the strands of DNA, so it's still held together, but it's broken. PARP comes in, kind of glues it together, gives you that repair function, and then it leaves when it's done. Right? And you go back through the process and you just keep replicating. So PARP's really important. What's interesting when you look at these different genes is that they actually work together. Remember how I was telling you the brakes will stop the car while the accelerator can still keep working? It's like the car can stay alive and keep functioning, metabolizing, but the brakes are holding it so it can't replicate yet. Some of these genes work together, like the BRCA and the PARP genes. And what they're actually finding is that if you have a mutation for BRCA is the brake, remember? PARP is the repair mechanism. If you have the mutation for BRCA and PARP works normally, then when the cell's DNA is broken, it may keep trying to repair it, but it may repair it flawed because there's no brakes giving it time. It's like trying to repair your car as it's driving down the highway. So PARP's trying to fix this car when there's no brake working. So it causes more and more mutations. So what they found is if they give people that have this BRCA mutation, if they give them a PARP inhibitor to stop the PARP, what will happen is that break in the DNA doesn't get fixed and it will completely rip, which means the cell will be determined to die. So it's kind of an interesting process. Then that cell dies, and then they just grow a new one that's a healthy cell into its place. So we can use those theories for treatment too. So always think this is really physiology. It's critical thinking. It's thinking out of the box. It's not one pathway is an absolute. It's a combination of the pathways to get you the right answer or the best solution. So next, does cancer usually happen after one mutation? I've already answered that a few times, and it's usually no. Sometimes it does, but it's usually no. Cancer is considered a disease of aging. It's because you acquire all these mutations over time and it's slowly changing. So you have this normal tissue here and you get a little bit of a mutation. So tumor suppressor is lost. Then suddenly, before you know it, you're making all these extra cells. Well, then you lose another, you lose another function. In this situation, you lose RAS. RAS is out of control now. So the brakes are gone and RAS is now stuck to this floor and it's completely replicating over and over and over again. So you get this rapid growth. Now you have this big polyp that's growing. Right? Then you get another loss of another tumor suppressor, which means that the polyps get even bigger and even faster. And then before you know it, you're losing an important gene, which is called P53. I'll show you in a few slides. This P53 is important because it repairs, it helps you use the brakes, it does all kinds of functions. But as soon as you lose this one, most cancers, if I remember right, 90% of all cancers have a dysfunction with P53. Once you've lost P53, this thing just goes crazy. It looks like a stem cell, it starts growing out of control, and then before you know it, you have full-blown cancer. So it's an accumulation of mutations, not just one. It's a disease of aging is why they call it that. Right? So one of two things happen for tumors. Either you increase the growth rate or you decrease apoptosis. When you lose P53, you've done both. You make it grow faster, replicate faster, but you also decrease its ability to die normally. So you have 100 trillion cells in your body, and every second 1 million of them die, which means you have to replace them at that rate. If you're not killing them and you're still replacing them in a million, before you know it, you're huge. You know, you're just blown up. That's what a tumor is. It's an area that's replicating faster than the cells can die. Right? And don't forget, every time you replicate a cell, it's like taking the entire Encyclopedia Britannica collection and replicating it word for word, letter for letter. So that's a lot of responsibility for a cell. Right? Your book uses this example. Like, we know the progression of some of these mutations, like for instance colon cancer. Here you have normal colonic cells, they look normal, and then you get this one. This one now suddenly is lost an enzyme that basically tells it to die. So it doesn't know to die anymore, so it just keeps replicating. Well, now those keep replicating, even with their flaws. Now you've got another mutation. Now the accelerator's stuck on, so it's accelerating faster. And now you're losing the ability to use the brakes. And then before you know it, P53 is gone, and you've got this full-blown cancer stem cell going on. Right? So there's a progression to it. You can watch the progression. That's why, since cancer is a disease of aging, after the age of 40, we have to get colonoscopies to see, do they see polyps? If they do, they'll take a little sample of it and they'll look at the cells. The cells look like stem cells, then they're malignant. If they don't, then they excise this polyp, they cut the extra tissue out, and then they hope that it doesn't grow. It's kind of the same idea with a pap smear. So you have normal tissue, you take a sample, look at the cells. If they look like this, you have to dig all of those cells out. Next, what prevents normal cell immortality, and we talked about this in genetics too, it's that telomerase chemical. So remember, the telomeres are the ends, the little caps, like shoestring laces where you put the plastic on the end to hold them together. That's what a telomere is. And remember, with normal cells, every time the telomere, or the cell replicates, the telomere gets a little bit longer. It's like a candle wick. You know, you keep burning it down, and the candle itself gets shorter, but the wick seems to get a little bit longer and poking out. And that's what's going on. Telomerase keeps that wick exactly the same length. Cancer cells find the way to keep that telomerase working. And then the last thing that cancer cells have to do is they have to feed themselves. They lose that social boundary, remember? They don't share evenly. They don't say, well, you get some blood and I'll take some blood and we'll share evenly. They want it all. So what they do is they release this stuff called VEGF. It's called vascular endothelial growth factor. This VEGF causes the endothelial cells to start growing, and they grow out. Here's your normal blood supply. If this is a tumor, it releases that chemical and tells these cells, come this way. And they start branching out over to the tumor, and they're bringing the blood to the tumor. So the tumor doesn't even have to move at this point. Now it's getting all this fresh blood supply. It keeps growing, getting bigger. It's starving the cells around it, which is going to eventually starve out the cells. They atrophy, and they shrink. And this tumor keeps getting bigger and bigger. Well, cancers do the same thing. And what's scary is that once the cancers get access to the blood supply and the lymphatics that come with it, then it can go anywhere in your body. So there are a handful of traits that actually are signs of cancers. And before I move on away from this section, I want to make sure that we talk about P53, because I kept talking about how P53 is so important, and it's kind of one of those distinguishing characteristics of cancers, and about 90% of all malignant cancers have a flaw in P53. Why it's so important is because it actually helps repair the DNA. It plays the role of a repairman. It also is a tumor suppressor gene, so it holds the cell. It keeps it from moving. So not only does it hold the cell, but it also does the repair. This can also initiate apoptosis. So if the cell can't fix, or P53 can't fix the cell, it'll help the cell to die. And then it also triggers angiogenesis. So it blocks angiogenesis. I said triggers, even the wrong word. It blocks angiogenesis. So in other words, P53 does all of these things to prevent this mutated cancer cell from growing, from, well, if it has a flaw, helping repair it, prevents it from living, so it helps it kill itself, and then it also prevents it from getting food. P53 is such a critical enzyme. It's definitely one you want to keep your mind on. Right? So the hallmarks of cancer, I kind of showed you this slide earlier, but we'll keep using it over and over again. So they become self-sufficient. They tell themselves to grow. They're autonomous, remember? They're insensitive to anti-growth. They don't listen to the surrounding cells. They evade apoptosis. They become immortal. Right? They have limitless replication ability. They can replicate or proliferate over and over and over and over again. They actually have a way to reroute blood to get into your tissue. So, and if you look, a lot of the cancer treatments target, it's stopping this angiogenesis, stopping that from happening. So they try and block that VEGF stuff. And then tissue invasion and metastasis. So these cancer cells can actually move from their original state. Right? So I've already gone through and I've listed a lot of these things that change in each of those areas. So you can see how the BRCA or the RB changes, and what it does is it turns off the anti-growth signals. So the tumor suppressors are lost. You can see how the accelerator is stuck on. You can see how P53, when you lose that, it doesn't go through apoptosis anymore. How VEGF will cause vascular growth. And then telomerase gives it that, well, infinite life. And then inactivating E-cadherins I talked about in the last video, but keeps them from sticking to the normal cells and they can move and travel through your body. Right. So here's just the last example, putting it all together when you look at prostate cancer. So I showed you pictures of breast cancer before. Here's a normal prostate. Right. If you look at the progression, here's the glandular epithelium of the prostate. Remember, the prostate releases enzymes into the semen to help protect the sperm. So it's actually an exocrine gland where it releases chemicals into the external part of the body. And then it's also endocrine because it releases hormones too, like prostaglandins. But here you can see the normal cells, you can see the normal boundaries. These are normal. But as they progress, you can see what happens. Things like inflammation, reactive oxygen species, telomeres changing. All those things in here start changing the cell. Right? We even know the genes. We know the genes that are involved that cause the changes. You can see epigenetic influence that cause changes. But what happens is slowly they start changing. And like I said before, you can see just by the way they stain extremely dark, cancer cells are like that. It's because the nuclear substance is staining so dark and it's so full in the cell that it makes it really kind of a bold color. Right? So here you can see prostatic hyperplasia. Here it's growing bigger and bigger and bigger in numbers. It could be hypertrophy too, but you can see all these little basically benign tumors growing all over. A typical prostate's about 4 centimeters across, and this one looks like about 6 because it's past the 5 centimeter scale. And then it's getting even worse. You can see how these cells are barely even distinguishable from the rest. And then it's just so bad that they're throughout. And at this point, this is where it starts metastasizing and moving downstream. But you can see the processes involved in changing it. You can see the different genes that are changing their expression, so we know that. We can actually take samples out of somebody's tissue or blood and find out what's going on in each of those changes. Right? So we're going to stop the video there and come back on the next one when we talk about gene and environment interactions.